Hello, we are going to talk about what happens at the synapse of a neuron. So last time we talked about the neuron, the different parts. We talked about the action potential flying down this axon, and then it reaches the axon terminal, which is a dead end. It needs to get across that synapse in order to stimulate the postsynaptic cell. This postsynaptic cell could be another neuron, it could be like the dendrite of another neuron, or it could be something like a muscle, like a smooth muscle, cardiac muscle, or even a gland, or even skeletal muscle. So what we'll do is we'll kind of zoom in on this synapse, and we'll kind of zoom in on this synapse right there, okay? This is going to be the presynaptic cell, or the axon terminal of the neuron that we're talking about, and this is the postsynaptic cell right here. This could be the dendrite of another axon or another neuron, or it could be a muscle cell. Okay, so this is the synapse right here. The synapse is super small in width. It's only 30 nanometers wide. And just to give you a sense of how small that is, you would have to line up 30,000 or so synapses side by side to get a millimeter in length. And so that's a very small in size. So first thing that happens is we're going to have this action potential that flies down the axon, and when it reaches the axon terminal, it's a dead end. This depolarization in the charge inside the axon associated with this action potential is going to stimulate some calcium voltage gated channels that are going to be situated in the cell membrane of the axon terminal. So everything in red, these are calcium voltage gated channels. Voltage gated means that they will be open or closed based on the voltage inside the axon. At the resting potential, which is -70, they are closed. When that voltage goes up during depolarization of the action potential, they open. So this is a calcium voltage gated channel, right? So the action potential reaches the axon terminal. These calcium voltage gated channels open when that action potential reaches that location. And there's a lot of calcium on the outside of the axon. That's what all these red dots represent. And so all this calcium is going to, not all of it, but a lot of this calcium is going to go into the axon terminal. Okay, that's the first thing that happens, is calcium comes in after these calcium voltage gated channels open. All right, I'll kind of draw it in a couple of different ways. Now there's something else that's going on inside this axon terminal. There's going to be a bunch of little spheres. They're like bubbles that are made up of the same cell membrane that makes up the neuron itself. These little bubbles are spheres. They're called vesicles. And these vesicles are going to house a bunch of tiny little chemicals called neurotransmitters. The vesicles are small themselves. They're only about 30 nanometers in width, so the same width as the synapse. And they're going to contain a bunch of tiny little chemicals, which I'll draw as these little purple, maybe not, these little pink dots. All right? These little pink dots are neurotransmitters that are located and housed inside these vesicles. These neurotransmitters were made way up at the nucleus, but they migrated all the way down the axon, and where they are stored and housed in these vesicles. There's a bunch of different neurotransmitters in our body. A lot of common central nervous system neurotransmitters are things like serotonin, dopamine, endorphins. Very common neurotransmitter in motor neurons are acetylcholine. This is the neurotransmitter that stimulates the contraction of skeletal muscles. Glutamate is a neurotransmitter which is often associated with the stimulation of sensory afferent nerves. So there's a bunch of different these little chemicals. Each vesicle is going to contain a couple thousand of these neurotransmitters. When calcium comes in, it's going to cause the vesicles to fuse with the cell membrane of the axon itself. And when that happens, it's like two bubbles touching, they pop together. So I'm going to try to get fancy here with white, right, with a white marker. But basically it's going to cause these neurotransmitters to fuse, or the vesicles to fuse with the wall of the axon, and that's going to cause this to happen. They fuse, and when they fuse like this, that allows all these pink little neurotransmitters to diffuse the short distance into and across the synapse. These neurotransmitters are then going to bind to a completely different type of channel This is called, in orange, a chemically gated channel. It's chemically gated because they open when they bind onto a chemical. So this is a chemically gated channel. Chemically gated channel, which is located right here. When this neurotransmitter binds onto the chemically gated channel, they open, and they're going to allow a couple of different ions to flow across. They're going to allow sodium to come in and potassium to go out. But more sodium is going to come in typically because sodium is, so I'm just going to label this orange, these orange molecules of sodium. All right, mainly sodium is going to come in. So sodium is going to be in red or in orange, and just to be consistent, calcium was in red. So these chemically gated channels open, a lot of this sodium is going to fly in. Sodium has a positive charge, and so that's going to cause this postsynaptic cell to depolarize, or the voltage inside that cell is going to increase. If we were to look at a chart that shows the voltage inside this postsynaptic cell, it's held at the resting potential at -70, just like the axon is. Okay, so it's going to be hanging out right about, let me see, what color should I use? We'll use green. All right, so this voltage is going to be -70. And then as sodium comes in, it's going to cause that voltage to go up because sodium has a positive charge. But let's consider a situation here. Let's imagine that only one or two action potentials came down the length of the axon. Now remember, the frequency of action potentials in axon determines the magnitude of that signal. If this neuron is really excited and it is encoding a very strong signal, it's going to be firing as many action potentials as it can, one after another. If it's a very weak signal, it's only going to be like one or two action potentials. Let's consider a weak signal. One or two action potentials travels down the length of the axon. It's going to cause a couple of these calcium voltage gated channels to open. A little bit of calcium comes in, which causes just one or two of these vesicles to fuse with the membrane, which allows, a couple thousand of these neurotransmitters to diffuse into the synapse. Okay, this is a vesicle, and these guys are in pink. These guys are neurotransmitters. Causes a couple of these neurotransmitters to diffuse across the synapse, which is going to bind to only a couple of these chemically gated channels, allowing a little bit of sodium to come in. It's going to cause a depolarization, but it's not going to be a very extreme depolarization. It might just be a little blip that looks like that before it goes back down to normal. That would be different if this axon was firing like crazy. Let's say it was firing with a bunch of different action potentials one after another. That would cause more calcium to come in, more vesicles to fuse, more neurotransmitters to fill up the synapse, more chemically gated channels open, a lot of sodium comes in. That's going to cause a much stronger and more extreme depolarization. These two blips on this chart, these are called graded potentials. Graded potential. These guys, the magnitude of the graded potential reflects the strength of the signal that was sent by the postsynaptic cell. Graded potentials are typically going to exist in dendrites and in places like, the sarcolemma of a muscle fiber, whereas action potentials are going to exist in axons. Sometimes chemically gated channels might open that allow negative ions to come in, like chlorine. If that happens, it's going to cause, chlorine has a negative charge. If negative things come in, it's actually going to cause the charge in this dendrite or this postsynaptic cell to hyperpolarize and go down. Graded potentials that go down, or hyperpolarizations, those guys are inhibitory. Whereas graded potentials that go up or are associated with depolarizations, those are excitatory. If this is a dendrite, an excitatory graded potential is going to tell that neuron to fire, whereas an inhibitory graded potential is going to tell that neuron to not fire. Now this is kind of where it all comes together. Let's go back over here to our picture of the neuron. And let's imagine that one of these dendrites is receiving a bunch of really strong excitatory graded potentials. All of these really strong graded potentials that it's receiving one after another are going to add up and combine on their way to the cell body to produce one really large depolarization in the charge of the cell body. That's going to cause this neuron to most likely fire. The fact that those strong excitatory graded potentials added up on their way to the cell body, that's called temporal summation. Spatial summation is completely different. Let's imagine one dendrite receives an excitatory graded potential and another dendrite over here at the same time also receives an excitatory graded potential. Both of those signals are going to travel towards the cell body. They're going to meet in the middle and they're going to add up to an even larger excitatory graded potential. They add up spatially from different places, so that's called spatial summation. Sometimes one dendrite might receive an excitatory graded potential, where another dendrite at the same time might receive an inhibitory graded potential. When those guys meet in the middle, they're going to cancel each other out. This and this cancel out. There's going to be no change in voltage, and this neuron is not going to fire. Okay? So this is a very simple way in which neurons can process signals, right? And all this happens at the synapse, right, where things connect together. So connections between neurons really allow these neurons to process information. If we were to kind of expand this out from not just one connection between two cells, but the millions of connections that exist in your brain, these connections or synapses really allow us to process complex information. This allows us to solve problems. This allows us to think critically and allows us to react to different things that happen in our world. Okay, so signals really can be changed or modulated or processed at the synapse, whereas the axon is really all about just sending a signal from point A to point B. All right, that's it. Thanks.